The Biofilm Center in Duisburg, Germany. We're beginning this project, the Alliance project, with several partners, which is involves the exploration for the possibilities of life and habitability in Mars. As is obvious in this talk, I'm going to give you an overview about what we do in Duisburg in terms of biofilms, something very general, and give some information of running experiments and planned experiments and what we expect to get from this collaboration. Why biofilms? Well, due to their size, microorganisms are highly surface active. They interact greatly with interfaces. Microbial attachment to surface is a very known phenomenon ever since the annals of microbiology. Already in the 17th century, Leon1 was describing bacteria attached to teeth and to other surfaces. Later on, in the 40s, Sobel did further studies about the influence of surfaces in bacteria. But very amazingly, the trend for years, what made microbiology famous everywhere, was to isolate microorganisms and concentrate in the so-called planktonic phase. This, in fact, brought most of the great advances in microbiology, from the discovery of antibiotics to many other advances, all speaking to a very, very limited part of the microbial world or the microbial cycle of life. Today's fact is that a high percentage of microorganisms, you can say that up to 99%, are not culturable with present methods. Only a small percentage of these can be isolated, and they gave the rise to the golden age, as I just spoke. And this was probably due to a lack of molecular biology techniques, and this made not possible to go into deep into complex microbial communities such as those forming biofilms. Another fact is that most of the presently known microorganisms on Earth live permanently, or at least spend a great deal of time in these structures. I just come with a formal definition that was proposed by Donald and Costerton in 2002, and this says that a biofilm is a microbially derived sessile community characterized by cells that are irreversibly attached to a substratum or at an interface to each other, or are embedded in a matrix of exopolymeric substances that they have produced and exhibit an altered phenotype with respect to growth rate and gene transcription. This definition is important to go away from the concept of a film. These are highly heterogeneous structures, just that it is embedded in the name. But it applies to both also films on surfaces, as well as to flocs, the so-called floating biofilms, and as well to mats and sludges, or the so-called thick biofilms. Characteristics that are common to these aggregates are that they're kept together and adhered to the surface by a matrix of EPS, extracellular polymeric substances. And these matrix constitutes not only microbial polymeric products, but all range of trapped and sorbed material from the environment that includes debris, corrosion products, soil particles, etc. Over the years, this is the model that has been proposed in terms of a life cycle of the biofilm, which means that people assume that now what we think of a planktonic phase is only a transitional state from one biofilm to another. Everything starts with the creation of a conditioning film that it is created by absorption of polymeric substances, by absorption of proteins and other material Then bacteria tend to attach, reproduce, and start producing the polymeric substances until you have a mature biofilm, which in turns with time will eventually release again bacteria that will continue the cycle.
Could you go back? I know it's just a sketch, but then 1, 2, 3, 4, 5, you have these pictures on the bottom. Is there a sense of the relative length scales? I mean, say, what's the horizontal length scale relative to the vertical length scale in a real active?
You mean like a time scale?
Physical? No, sorry, length scale. Size. Length scale.
Size. Yeah, no, it's completely not in scale, of course. It's
okay, but if this is the cycle that happens in a real biofilm, okay, over what length scale do you go from the number 1 to number 5? What's the
micrometers? So you mean, yeah, micrometers, and here you cannot think of a space difference here. It's maybe the same structure, and what happens on the formation can occur in other structure. It's just a generalized stepwise process that can happen, but it's not. You cannot think of a length distance here. That's not there.
But there is a thickness to the thing?
Yeah, micrometer. It can vary very much from species to species. Well, it's very important in terms of the first phase of this structure, the attachment. And from this comes the variety and the huge possibilities on cell surfaces. Here I just wanted to bring some examples of the heterogeneity of these surfaces. They can go from single appendages, this flagella, to multitude of flagella to pretty much naked structures such as this. This is Methanosarcina, I think, cocci. They, of course, confer different attachment properties, and here this is a little bit borrowed from the colloid interactions. With bacteria, you will have that there will be certain difference depending on the appendages that they have. Particles will be limited by this electrical barrier in surfaces, but in some instances static interactions can be important in terms of attachment. In the same sense, there can be the so-called shadow or blocking effects, where depending on the shape of the microbe, you can have avoiding of attachment due to blocking effects. For example, in porous media, in porous material, this can be quite important in the sense that some bacteria that are already attached can create repulsive effects that avoid other bacteria to be attached. And this is quite important in terms of the first stages of this biofilm formation. As I speak before, there is a conditioning film that is composed of polysaccharides and proteins that are a surface within minutes of liquid contact will be covered by these substances, and they indeed induce the attachment of bacteria, sometimes with the breach effect of, for example, cations, especially divalent cations are very active in interacting between the surface and the bacterium and facilitating the attachment. These extracellular polysaccharides and proteins, these EPS, are normally loosely attached to each other, and that's one distinction from more tightly configured capsular polysaccharides and lipopolysaccharides, which are considered to be part of the cell itself. This is a little bit of controversy where the biofilm starts. We have examples where this capsule, in some literature, they just put it as a feature of the cell itself, or in other cases it disappears from this. They only give the information until the outer membrane. But we can see that these capsules can be quite, comparing with the size of the bacterium, can be quite big, and can be quite probably be considered part of the extra polymeric substances. So this is the question: where do we speak about the interface between the bacteria and the environment? Does this include the EPS or not, or until what extent, and which would be the influence? The second phase after the attachment, the heterogeneity, is EPS production. EPS is basically, so in terms, the biofilm will be composed 99% almost of water, and in terms of the EPS only, the exopolymeric substances, about this varies very much from biofilm to biofilm, but generally 50% carbohydrates, 40% lipids, and only around 0.5% proteins. This is atomic force microscopy of highly hydrated biofilm examples. So they're highly hydrated polymers of microbial origin in which biofilms organisms are embedded. They contain both soluble and insoluble polysaccharides, proteins, nucleic acids, and there are many substances coming from the environment that is also considered part of the EPS. They fill and form the space between the cells and are responsible for the morphology, which in turn affects very much the metabolic and physiological characteristics of the containing organisms. It is the immediate environment of the biofilm cells, and indeed determines their living conditions. So here is just some examples of the typical monosaccharides found in biofilms. They're both homopolysaccharides, heteropolysaccharides. It's very common to find acidic substituents. For example, acetylation marks a very important distinction between the morphological characteristics of biofilms. Especially well studied has been the case of Pseudomonas aeruginosa. Strains that are mutants that are lacking the capacity of or the acetylation in the polysaccharides produce completely different morphology than acetylated counterparts. Alginate is indeed a good example, but it's not representative of environmental biofilms due to the fact that they variate almost from species to species, so it's not applicable to real life examples. This matrix is sustained by a wide range of weak interactions within, we can say, hydrogen bonds, here the bridging effects with divalent cations, and also it can contain repulsive forces that of course prevent the matrix from collapsing and gives this sponge type of configuration. They can have different architectures, and this brings back the characteristics of the flow regime and on the environment where they're developing. There are usually when there is turbulent flow, they form this streaming type of biofilms, which are like hairs going with the flow. They can have a rolling type of behavior, and something very important is that depending on the surface that we're speaking, they develop either thick, continuous biofilms, or very patchy, sketchy biofilms when it's in the case, for example, of porous media, for example, in soil or in sand. This is an example of a quartz sand, and how they grow only in the pore throats of the sand, but it's not as continuous as we would expect from a flat surface. So the concept of film can be misleading, of course, when speaking about biofilms. It's very difficult to identify a real interface here. You will have places in the biofilm where there is practically no interface. There will be channels where different transport regimes are happening. So in terms of applying a model here, it's extremely difficult because you cannot generalize and say there is just a film covering a surface.
In this picture in the upper right-hand corner, is it known why the film forms in a certain place? If you were to scrape off the surface of the sand and let it grow again and grow again, what's the aerial coverage? Why is it known why?
Yeah, of course we're not physicists, but it has something to do with the different flow regimes that you can create in the porous media. So in some regions where they are more exposed, the velocity vectors will be higher, and probably that will decrease the possibilities of formation. And in places where the flow is less, or there is stagnant points, you'll have a bigger chance of cells to attach. And that also plays a role, the motility, if they are motile or not, that also plays a role, and the saturation state of the material. So if you have completely saturated particles, then the size of the liquid phase will be higher, it will be bigger, and they will have more distance to travel up to the surface. But when it's unsaturated, this film of liquid will be thinner, and they will have more possibilities to approach the surface. That all this influence the possibilities for attachment, and where do they form, and where they can prosper.
But has, for example, anyone done an experiment where you scrape off this surface and impose a regular flow, say a shear flow, and see if there's a pattern that's different than in the absence of the shear flow?
Yeah, there is studies of this, and this is Paul Studley in Montana, and he has done very much studies with this type of trying, for example, laminar regimes flows, or turbulent flows, and observing what happens with the formation of the.
If you're looking for the literature on that, Paul's work is on teeth, biofilms on teeth. Quite often, a lot of that kind of work is done on that. He's funded by the toothbrush companies.
I see. Well, then it's only turbulent flow.
Well,
both.
I mean, there must be a correlation with the roughness on surfaces. I would think that the biofilms are still sort of micron, so even if you had a turbulent flow, you may well be within the boundary.
The viscous boundary layer is small relative to that.
Yeah.
So, I mean, 10 to the minus 6 divided by the velocity, whatever the velocity is. So the viscous sublayer is small, so so the roughness is crucial. So that's really my question, whether or not the green that we're seeing is simply associated with roughness, or it's associated with something else. That's
what we have observed. I have a picture after a SAM picture, that they really form in places that are not exposed to the flow. So in rough places where there are sort of caves that they can be there, and there you can find most of the material. Here is this is an example. This is the sand grain, and here you can find bacteria, but when you look at the content, organic content is not as high as when you have a flat surface that is continuous and you can grow them in there. They refuge in these places where are quite hidden from the flow, and that's what. But that doesn't mean that they don't affect, for example, the hydraulic conductivity of the medium. They increase it very much if they concentrate in pore throats, where they can drastically reduce the hydraulic conductivity, even though they're not in high amounts. Another aspect important is that these structures can be a complete microenvironment in itself, and they can be gradients between anaerobic and aerobic habitats. And you will have, for example, aerobic bacteria developing on the surface, and a wide range of redox processes going up to the top, where you can find practically a very nice example of ecological significance of these structures. So they're not single species structures. They are composed of a wide range of bacteria, which depending on the place that they are sitting, they're performing different physiological and metabolical activities. So that's quite interesting and quite important. It's not a homogeneous structure in terms of physiology either, or metabolism. Here is the example of the influence of acetylation in the EPS from Pseudomonas aeruginosa, SE81. This is the type. This is obtained with confocal laser microscopy. You can see that this mushroom type of structure is not found when acetylation is lacking from the polysaccharide. So this is also to take into account. So in terms for astrobiology, what we intend to do is how can these structures be protective in a harsh environment? What do we mean by protection? As a nutrient reservoir, perhaps against starvation for long times of periods, as a gene pool against environmental challenges. These places are places of very active gene exchange. So bacteria really learn in these structures how to degrade biocides, how to obtain energy from other compounds, etc. It's a very active environment for the close proximity that these organisms are subjected to. As a water reservoir, so these matrices are highly hydrated. Can they be a water reservoir? Can they have some effect against desiccation? And just as a physical protection against biocides, this is known. Bacteria in the human body, when they're embedded in biofilms, are highly resistant to antibiotics. Desiccation, shear stress, they are attached strongly when they're in the structures and when not. Pressure, UV, etc. This especially when they're in close proximity to minerals, so they can interact, they can trap, and could offer some protection. In terms of desiccation, the removal of water causes a stronger binding force because more binding sites can interact. In the drying process, here an example, the biofilm curls up and reaches parts of the mortar. This is just an example of a photosynthetic bacteria in the mortar. They respond differently to hyperosmotic stress. Genetically, they start the formation, or they code the genes to formate compatible solids, which are produced inside the cell to equilibrate osmotic pressure with the outside. When this happens rapid, they have not the chance to do that. When it happens slow, they have the chance and to make the bacteria survive. This is only at the bacterial level. Also for hypoosmotic stress, or when the solutes are released, they can resist low ionic concentration environments. And all these are a sort of tools that bacteria have to couple with this type of challenges. Yes,
maybe just a point of clarification, and I may be the only one wondering this. So are the biofilms precursor just biomolecules that attach to the surface, or are they things that the bacteria produce once the bacteria are on the surface, or is it both of these cases? For example, in these responses to changes in solute, are the bacteria producing more biofilm that help it adapt to the salt concentration gradient, or what's the
no? This is considering the cell itself. It's a direct response of a cell in a liquid, so it has nothing to do with the biofilm. It's in this state, it just responds to the environment itself, regardless where it is embedded. But both are these substances are both produced by the bacteria after attachment, but they also require some conditioning film that they have a reason to attach, to come to. So when a lot of nutrients and polymeric substances tend to attach to the surface, and this attracts bacteria to attach as well.
So in the case of these rough surfaces, I should think of biofilms in the fluid attaching to different parts of the rough surface that then promote colonization by bacteria, which then produce more biofilms in those. Okay.
But okay, again, so in that scenario, I have some baseline of EPS, for example, on a substrate. The physical scale of that, the length scale of that, is submicron, because we've established the biofilm as micron.
Oh yeah, that would be submicron.
But it's definitely submicron. Yeah. Because it's just polymer. I mean, is it a monolayer polymer or?
It is said it's a monolayer. Yeah. And then.
I mean, it doesn't have to be polymer. Anything organic is fine, I guess.
Correct.
Start with, I mean, when we try to clean surfaces, I mean, it's a really hard job to get rid of this stuff.
Yeah.
I mean, you leave a surface somewhere for 10 minutes and it's a piece of dirt with all this organic stuff on it.
Here speaking more about this EPS skin formation, bacteria when subjected to desiccation conditions are buried in an EPS layer, which prevents desiccation and helps them to survive to this environment part of what I was speaking before. There are examples of organisms that are very well adapted to desiccation rehydration cycles, and there is an extreme case, for example, of cyanobacteria. These are desiccated colonies of commune. As soon as you have hydration, they become completely hydrated, and they can and this is part of a life cycle, and it's very interesting that they have this ability. So what about potential Mars habitats? In terms of physical environments, well, Mars surface, we have the problem, very low water activity, low temperatures, influence of radiation. It is better to concentrate in parts where we have not so low pressures, a little bit higher temperatures, and this will occur mainly in the subsurface of the planet. Before, it was thought that life was extremely fragile, and examples here in Earth have shown that they can really thrive in places that are just some years ago not thought of. We're trying to, for example, in terms of physiology and metabolism, the involvement of iron-sulfur based metabolisms. Why? Well, there is lots of iron and sulfur at different oxidation states close to the Martian surface. And here on Earth we have the example of simple metabolisms, for example the chemolithotrophs. These organisms are able to obtain energy very without the need of any organic compound. They can do it directly from the oxidation of inorganic compounds. And most of them are also able to fix carbon for survival, so they're autotrophs as well. Most of them even are strict chemolithotrophs, so they are inhibited when you have organics present, something that is nice to think, because every time people think about growing bacteria, they believe that there had to be something organic on there, and that's not the case. Even though these bacteria need a lot of iron to produce, they need to oxidize a lot of iron in order to prosper, but even though several of these species produce biofilms, and that's quite a astonishing fact due to the fact that it's not an easy task. You need a lot of energy to do this. So we want to concentrate in iron-sulfur biofilms. They exist. This is an example of acid-tolerant biofilms. They colonize deep underground mines on Earth, for example. They are extremely acidic, almost battery acid type of pH, where they have a lot of dissolved heavy metals, for example, from dissolution of metal sulfides In this case, these iron oxidizing bacteria grow on the surface of pyrite, so iron sulfide minerals. Archaea dominate these biofilms, and they contribute to pyrite dissolution and the production of acid main drainage. So we have two options where in the study of these systems concentrate in this type of biofilms. They're more relevant for possibilities in Mars environments. And on the other side, also take the mucoid example that are not representative, but perhaps can tell us a little bit more about the possibilities for water retention and so on, these more thick biofilms. The biofilms that we can produce from iron and sulfur bacteria are very, very thin, in the orders of almost monolayers, but they can be interesting in terms of probably resistance to harsh environments.
So just to clarify, so both archaea can do just the same thing as bacteria then as well, creating these biofilms.
Yeah, probably the thing is that probably because these are mixed communities, it's probably that they're not in charge of producing the polymeric substance, for example, but that they can live in these environments as a symbiotic interaction that can also happen. Now I come to what we're trying to do. We have two species. We have observed something quite promising with them, and it's the fact that without one of species, when we have pure cultures of Leptospirillum ferrifilum, it's an iron oxidizer. This was isolated from an old iron mine. The organism actually can attach to pyrite in presence of pyrite, but it cannot produce thick or significant biofilms. But when they're in presence of a sulfur oxidizer, in this case we're using the Aciditiobacillus caldus, S2, they are able to produce aggregates in the planktonic phase and also biofilms. So we try to start experiments with microcosms, very simple, with, without pyrite, and with pure and mixed cultures. The idea will be to obtain the two extremes, in which in one case they're not surrounded by a great amount of EPS, and in the other case where they are, and exposing them to Mars conditions to see and to evaluate the survival rate of these microorganisms. That's the main idea. This is just we have with, without pyrite. We make a porous phase where by increasing the surface area we can have more chance for attachment. For these are pyrite crystals of 500 to 1000 micrometers. So we're doing microscopy on them. Sulfate and iron are just indicators of leaching from pyrite, so dissolution of pyrite, so just products from this dissolution. AFM to observe morphological characteristics of the biofilm. Then chosen systems will be exposed to Mars conditions, and finally we'll assess the survival. One idea would be to re-inoculate to fresh microcosms and see if they can again be active. And other vitality test. For this we have mainly fluorescence-based techniques, microscopy techniques, which I will speak after. In terms of, we have also a model mucoid biofilm. This organism produce thick mucoid biofilms together with the non-mucoid mutant. And with this we will make experiments with flow cells, in which we will try to observe directly with the confocal and laser microscope. These are dynamic flowing systems. Basically we will try to assess a growth medium with different water activities. That means just increasing or decreasing the salt concentration. And probably no growth medium for increasing periods of time, so eliminate completely water. And then observe what happens to the structure and to the survival of the organisms when we rehydrate. the biofilms. So what is the responses in terms of survival and structure? This is another part of our group that is just assessing the water retention in different alginate types. This is just because this bacteria produces high amounts of alginate, and they're taking commercial alginate comparing with bacterial and raw EPS, which means with the whole amount of polymeric substances. Until now, they haven't found any difference between these two, and they're continuing with the other. This is diagram of the flow cell systems. Here are the it can be an open system or recirculating system, and it was just what I spoke about.
But the test section there, you're controlling its temperature and humidity?
Here, no.
No. So this is just to prepare them for under more.
Yeah, they will be completely hydrated,
Yeah.
so under a flow, and then without or increasing salt concentration, but not to this, and not in a, well, the variation is not so high of temperature, but we don't control that.
And the range of salt concentrations is what?
Yeah, that can be from deionized water to 0.2, 0.3 molar NaCl.
These experiments are done at what temperature are they done?
At room temperature.
They're done at room temperature.
Yeah, room temperature. Okay. We use for the fluorescence detection with the microscope. We use basically to test vitality. This commercial kit, it's called Leon1 kit. It's composed of two types of fluorescent stains. One of them is called Cyto9, and it's able to penetrate all type of bacterial cells. When excited, it fluoresces green, and propylium iodide penetrates only damaged cells, so that you can and it fluoresces red. So basically, when you can see if they are intact when they're green, and if they are damaged when they're red. Of course, it's there are more fine tests to do this, but at the beginning we're trying to do that. There are lectins that have a special affinity to polysaccharides, and they can be fluorescent also, and they can be used to visualize polysaccharide fractions of EPS, so that you can probably detect any change on the structure, because polysaccharides are the one that determine more the morphology of the biofilm.
And is there any structure to the substrate you're using, or is it just a flat slide, or in these experiments?
Mm-hmm.
Is there any
they are flow cells that are just square capillaries, and they are connected with tubing. They're connected with tubing, and then in these flat capillaries, they're optical grade borosilicate glass, and we have no problem to observe the formation. Okay. What I just spoke about. IC analysis for sulfate and other sulfur species in terms of detecting any changes on the metabolism of the species. Analysis of iron as well for the same purpose, and as I said before, AFM for morphological features. This is some of the results we have obtained. This is just to notice the contrast between pure and mixed cultures in terms of attachment. These are experiments in short time, just observing the percentage of bacteria that attach to the pirate grains, and we have observed that in mixed culture they attach also, not as high as in pure culture, but one organism is completely not able to attach during the whole period of time. It is suggested that probably they can have some symbiotic interaction in which the elemental sulfur that is produced from the iron dissolution can be used by this microorganism as an energy source and can prosper as well. So this brings up the importance of symbiotic interactions when speaking about biofilms. This is a production of sulfate as well. In pure culture, there is almost no activity. When they're mixed or in pure cultures of the other organism, you can see activity. And here, this is the formation of bacterial aggregates after several days of interaction. Important to notice here is that in pure culture, they don't form these aggregates. So it means that they only do this when they are in presence of the other species, and that can be significantly important in terms of the survival of each species. We have said, for example, when we have a pure culture of Leptospirillum, there are no. This is also a fluorescent stain to detect all types of bacteria, called DAPI. And in presence of the other bacteria, the Acaldus, 10 times less concentrated than the Ferrophyllum, still you can see the two types of bacteria. Leptospirillum is very, very small compared with the Aciditobacillus, but you can see no aggregate formation. When you increase this to 5 times less concentrated, you start to see aggregate formation after 20 days of contact and in pirate. And at the same concentration, you can start to see huge aggregates, which we think are composed of a great amount of exopolymeric substances. So the idea is to have pure cultures, expose them to the conditions in Mars and in the presence of the other species in which they are probably forming or producing more EPS, to see if there is a difference in survival rate for both species. This is looking at these aggregates by fluorescence in situ hybridization, in which a specific probe for Leptospirillum ferrophyllum was used. So part of the DNA for a ferrophyllum that is known is made fluorescent, and it can be detected. And here we can see that probably much of these aggregates are dominated by this single species. That's quite interesting. We have to look further to this. This is an example of the life and death kit in relatively healthy systems. A few cells are dead, but mostly are active and alive. After being exposed to the conditions, we can hopefully see how the survival rate of these organisms. This is an example of AFM. Here is important the difference in the morphology. For example, here are aggregates from the planktonic phase. They have good resolution from the cells, but in the case of attached to parrot, there are what appears to be formation of polymeric substances. So that's also another part of the work we're performing there. So in pure cultures, there is no aggregate formation, and Leptospirillum ferrophyllum is able to attach to parrot and shows leaching activity. Aciditobacillus does not attach to parrot in isolation and does not induce parrot dissolution. But in mixed cultures, the picture changes, and we have aggregate formation, which increases with increasing concentration of Aciditobacillus caldus. There is attachment to parrot. And AFM suggests, as in the last pictures, an increment on EPS production of the attachment to parrot. This, of course, needs to be verified. The main message here is that complex microbial transformations with ecological relevance cannot be studied realistically based in isolated microorganisms. So it is just not the sum of the parts. It's not taking one isolate, studying, seeing how it responds to harsh environments, and then putting this back to conclusions. And the same applies for resistance adaptations. Biofilms are, of course, dynamic communities composed of a great diversity of species, and they are act as a biofilm as a whole, and this includes both biotic and abiotic components, for example, polymer cell, minerals, etc. And these studies of single species resistance and adaptability can be informative, but might not bring answers to habitability in other planets. So with that, that was the.
So the talk is now open for discussion. Any questions? Yes.
You had this black box that says exposed to Mars conditions.
Yeah. Precisely because we have to still speak with the other partners to see which are the exact conditions that we're going to expose them to. But assuming something relevant to Mars, low pressure, low temperatures, low water activities, and so forth. But that's
do you have any sense at all about the rate at which you might do that? Because that should be very important. If you take something and you quench it by 100 degrees versus slowly changing the temperature, that would, I would imagine, have a serious.
Yeah, well, that's part of the adaptability also of the systems and.
Well, on this kind of biofilms, there are no tests done anymore. But we have another kind of a more developed biofilm, for example, like we have made tests. And the temperature is in this case not really a crucial thing. If you are just putting these organisms and shocking them with freezing, where also EPS is present, there is no effect on the organisms. They are still alive and they can have a physiological activity. We have measured this.
So they're just like lawyers.
Sorry?
Just kidding. Okay. So is there any measurement of the osmotic pressure inside the biofilm for conditions in which you have well-known salinity outside of the biofilm?
Yeah, well, I'm not aware of this.
I'm pretty sure there is, just like in the algal physiology, these osmotic pressures, and bacterial physiologists would have made these measurements. I'm sure the Australians, Barman and Co. would have done this kind of stuff, because they're working on osmotic reactions of organisms being put into different salinity waters, and one of the crucial measurements they will make is internal osmotic pressure. So my guess is in the literature that data exists.
You just don't know the number.
Are there any observations or so of biofilms in permafrost, terrestrial permafrost?
Not described as such. There are assumed that they attach to surfaces and that they grow in this form, but the studies are centered in metabolic and physiological responses as a whole, but not as the structure. I haven't seen this.
So you don't have cultures from permafrost?
No. That's from the group of Wagner.
Are you prepared to say anything? I mean, because I don't know very much about it, about the communication between cells and biofilms. I didn't know that they do communicate to each other. So actually,
yeah, there are these. They produce this signal molecules, these homoserine lactones or so. I don't remember exactly the name, but they're quite important in terms of adaptation and how they will respond to different trends in the environment. And because they're in such close proximity, the molecules that are produced can be trapped in the EPS and can be detected by other species and can have this type of influence. Homoserine lactones are the name of these molecules, signal molecules.
Yes, I would like to have make a few words of comments. to relate that to our activity in this so-called alliance. This is one of the aspects we are dealing with there. And we have asked the colleague from Düsseldorf, from Duisburg, to come or to join the alliance, because we have the hope that that might be of relevance also for us in more general in the question of habitability. The title of the alliance is Planetary Evolution and Habitability. And so we think that for more not so life-friendly conditions, biofilms may be a very interesting aspect. And with respect to your question, what he has presented now is the result. I think it's the first step to see which of the cultures might be of interest or can be taken into consideration later. We already have performed experiments, not with biofilms. We are starting in our Mars simulation chamber, and we will torture them also in future in that chamber to study their survival or their active life, whatever happens at sea. So that is one of the very, I think, challenging aspects of our alliance.
So in that context, I'm wondering whether you have already looked at the rather challenging claims by Salisbury on the dark spots on Mars.
Yeah.
You have looked at those claims, or you know anything about those?
I have dealt with these dark spots from another point of view, not from a biological one.
Yes.
From a more or less physical point of view, because we see also, I will discuss it in my presentation
Okay.
also, we see some rheological or flow structures, whatever it is, and in that context we have dealt with. But not in the biological context. I think it's rather optimistic to think that it could be life behind it. It cannot be excluded, but it's in the polar regions or near to the polar regions, so it's rather cooler than on an equatorial condition. We expect to be more favorable for that.
Yeah, right. Good.
But if I may, just extending this biofilm stuff to the sea ice that I was talking about yesterday, it is our view of a large number of us that actually these brine channel systems are lined by biofilms rather than the organisms being free floating within the brine channel system. It's just that we don't have any mechanism by which we can observe this yet. But my guess is, I neglected to say in the talk yesterday, that the impression that we have is that it is a biofilm system that is filling these brine channel systems.
I could say in the laboratory we have done that. We've done solidification experiments in the presence of an EPS solution, and we can say that they do go to the surface. So even the whole suit with all the dissolved organic matter, you put it in contact with the ice, it goes to the surface, and it changes the surface energy, and we can tell that in a number of ways. So we certainly know in the laboratory, under conditions that are consistent with what happens in the Arctic, in that temperature and salinity range, that the EPS does form a biofilm on the surface, and it changes the energy of the surface. It's not published. I can show you pictures of it, but it's certainly true that from a thermodynamic standpoint, it would rather be on the surface than in the solution. So ice is more kind than the water.
So it's better in an Antarctic or Arctic snow or deeper ice than in ocean caused ices.
Yeah,
drier ice.
So that it might be a reasonable question to say, if you have ice in a porous media in a setting like Mars, then it may in fact be more reasonable for the system to form a film of water. That's why I asked about the osmotic pressure, where the biofilm would persist rather than on a cold, dry substrate, for example.
Something quite critical also on Earth is the movement of material from convective movement of material, that we can have a distribution of redox potentials. When you have a distribution of redox potential, you can have life regardless of any other limitation, basically. And that's the question: how mobile is this fluid in these conditions? Is it quite static? Is it what happens? Bacteria produce substances they need to get rid of. If they cannot get rid of that, then it can be that successful or not. That's the question. How is the convective movement in these confined spaces and in this porous media? And I think for life and for ecological terms, it's really, really important.
So, if there are not any further questions, let's thank our speaker again and reconvene at 15:30.